Right. Well, welcome, and thank you for joining us today for the 6th Paul Bertics Memorial Lecture hosted by the Biomolecular Chemistry Department. So Paul Bertics joined the UW faculty in 1986 and was the Kellett Professor of Biomolecular Chemistry at the time of his death. Paul's research program focused on understanding the regulation of cell proliferation and function by growth factors, cytokines, and bacterial toxins. And Paul excelled in research and in teaching. He received numerous awards for his research and was a member of several editorial boards and served on international grant review panels. As an educator, Paul received the UW Chancellor's Distinguished Teaching Award and many teaching awards in the School of Medicine and Public Health. Each year, we honor our colleagues' memory by hosting a scientist at the forefront of innovation and excellence. And we are delighted that Dr. Cheryl Arrowsmith will give this year's Bertics Lecture. So Dr. Arrowsmith received her PhD in chemistry from the University of Toronto and carried out postdoctoral research at Stanford University with Oleg Yardetsky. She is a professor of medical biophysics, senior scientist at the Princess Margaret Cancer Center, and holds a Canada Research Chair in structural genomics at the University of Toronto. Dr. Arrowsmith is also the director of the Toronto node of the Structural Genomics Consortium, or SGC. Founded in 2003, the SGC is an international public-private partnership focused on elucidating the functions and disease relevance of all proteins encoded by the human genome. The SGC has contributed thousands of high-resolution protein structures and over 130 chemical probes. And the SGC has distributed over 25,000 samples of these chemical probes to the scientific community. So Dr. Arrowsmith's interdisciplinary research program studies the structure and function and chemical modulation of proteins involved in epigenetics and nuclear signaling. And her lab uses structural biology and chemical biologies to understand how transcription and chromatin regulatory proteins recognize, interact, and signal to the molecular components of the cell. And in conjunction with SGC, her lab uses the protein structural information to develop new chemical genetic tools to define, perturb, and manipulate the essential functions of proteins involved in chromatin signaling. Dr. Arrowsmith has served as a member of many scientific advisory boards, grant review panels, and journal editorial boards. She is a recipient of many research awards and was named a Fellow of the Royal Society of Canada and a Fellow of the American Association for the Advancement of Science. So please join me in welcoming Dr. Cheryl Arrowsmith for the 2022 Bertics Memorial Lecture.
Thank you, Peter. Everybody hear me? It's a real honor to be here and to give the Bertic Lecture this year. I didn't know Paul Bertic, but I know he was a valued colleague and amazing scientist and teacher. And so I hope I can live up to the expectation of excellence in my lecture that I will tell you about in a minute. But I thought I first read over some of the read through the publications of Dr. Bertic to understand his, his research, and I found a few threads of commonality between what I do and his research, and so I thought I would just mention them ahead of time. He worked on glioblastoma, GBM, and I'll have a section in my talk about our project on GBM. He also worked at the interface between cancer and the immune system. And I also have a section or a project where we also have run across that interface. And interestingly, there was a flash from the past for me when I read about his work on liquid crystals and symplectic phase liquid crystals. He was using them in a biological context, but when I was an undergraduate senior thesis student at Allegheny College for my senior research project, I also worked on liquid crystals, trying to get an X-ray structure of them. We never actually got to that stage, but I did work with liquid crystals at that point. So I'm glad Paul got much further than I did in that area. So, thank you, Peter, for the introduction. we'll tell you about our work, both in my lab at Princess Margaret Cancer Center, the University of Toronto, and the Structural Genomics Consortium. And let me start with kind of the big picture of how we are thinking now about the genome and the 3D structures of the proteins encoded by that genome, and how the importance of these proteins on a genome proteome-wide scale for human health and disease. And this figure illustrates one of the problems and the challenges in leveraging the potential benefits of genomic research, where now we know what all the genes are in the human organism, and we can predict what the proteins are that are encoded. But if you look at the numbers of publications, so this graph along the x-axis is a gene or its protein ranked by the number of publications versus all 20,000 of them. And the top ranking one here is P53, and then the rest you can see the very small proportion of the genome and the proteome has large numbers of protein studied. And this graph looks the same as it did in roughly around 2000. So one of the problems here is that high rate of clinical failure, especially for new protein targets. When a new drug is made to a new protein target, very often the drug fails in the clinic, not because of tox or other issues, usually because of lack of efficacy, meaning we didn't choose the right target for that disease. And it means we don't understand human proteins or even human biology well enough to know what the right targets are for a disease. And this is related to this vast dark proteome, we think. If so much of the proteome is unstudied, there's an awful lot going on out there that we don't know about. Okay? But if you look at unbiased genomics, and in the case of genome-wide association studies, for example, SNPs associated with disease, or the OMIM database of 1 million inherited genes involved in disease, you can see that almost every protein or gene has some disease related to it. And we view that this dark proteome harbors a lot of potential new drug targets, yet they're not very well studied. So we think this is this is an important area to study. It's hard to get grants to work on a protein that nobody else has worked on, with not much is known about it, right? So our consortium that is funded by a consortium of pharmaceutical companies is one of the types of strategies where we can work on the unknown and also the known when there's a clear problem to solve. But this allows us to try to address some of these gaps here. So when we started about 12 years ago, transitioning from structural biology to characterize the proteome to chemical biology to characterize those proteins that regulate those proteins in the proteome, it's a humongous challenge, right? So we thought we would focus in the area of epigenetics and chromatin regulation. It was an exciting new focus for drug discovery at the time, around 2008, 2009. The work of David Alice and Peter when he was in the lab, and many colleagues was a very, very exciting time. It still is. So we thought this was a good area to focus our efforts. So just a little bit of background on epigenetics and how we're thinking about it. Our genome has, as an individual, a single genome with DNA sequence, but the way in which the genes are expressed, turned on or turned off, are dependent on the chromatin state, post-translational modifications on the protein histones, and remodeler proteins, and also methylation and other marks on the DNA itself. And so it's this combined sets of modifications and proteins interacting with chromatin that determines whether a specific loci is expressed or not. And these are organized so the epigenetic and chromatin organizes the genome for expression into specific cell types. And this is how we get from in a single individual cell to all the differentiated sorts of cells. It is an epigenetic process development. And it's also the case in disease. For example, cancer. The epigenome in cancer is almost always aberrant and sets that cell type identity, which is often the cell type identity in cancer cells is disrupted. But epigenetic states can be reversible under the right conditions, and so as opposed to a mutation in the DNA, which is not possible to reverse. So I work at a cancer institute, and much of the epigenetic research, especially with respect to drug discovery, has focused on cancer. And we can go through almost all the hallmarks of cancer and identify ways in which epigenetic changes are relevant. So frequent mutations in chromatin regulators, altered DNA methylation patterns, comprise DNA repair and genome integrity, cellular states that block differentiation. I'll show you a vignette from our research that illustrates this. Cellular states that enforce altered metabolism. That's a very ripe area for investigation. Establishing and maintaining gene expression programs that can drive proliferation. These cancer-specific epigenetic states can create a targetable vulnerability in certain cases for the creation of new drugs. Another way to think about it is that the gene expression program in a given cell was just determined by its epigenetic state, is dependent on the transcription factors and chromatin remodelers and regulators that drive those transcriptional states. For a long time, the cancer field has been interested in trying to drug aberrant, overexpressed transcription factors, which are known to play an important role in many diseases, especially cancer. But these tend not to be very druggable. On the other hand, the chromatin regulators on which those transcription factors depend tend to be druggable. So we and others over the last 15 years or so have shown this using structural biology and chemical biology. So this is the area I'm going to elaborate on in the rest of my talk. So this is our consortium program to make chemical probes. So what do I mean by a chemical probe? So here we mean a synthetic chemical compound that is a probe of the activity of the protein of interest in a cell, of a specific protein of interest. And so these are drug-like small molecules that selectively usually inhibit, but in principle could also activate a protein activity in the cell. And then they're not very easy to make. It's the very early stages of drug discovery. When the drug makers in the pharmaceutical industry want to target a protein, this is the first thing they make. They make a selective inhibitor to the protein of interest, and then they take that compound into disease models and ask, is inhibition of that target modifying my disease phenotype in the fashion that we want? And this is why our consortium, which involves now 8 pharmaceutical companies and over the past years has included a couple others, is interested in helping us make these cofactors. In fact, they do most of the medicinal chemistry, and we do the protein work and the cell biology work to come up with these potent, selective, cell-active compounds. And we also characterize them for not being broadly toxic and having off-target activities. Okay, so they're highly complementary to genetic methods, such as knockdown or knockout strategies. They're compared to genetic methods. They have an ease of use in multicellular systems. You can do temporal studies with them, and they're closer to the therapeutic modalities. So they certainly don't replace genetics, but they're highly complementary. And we make these fully and fully available to the research community to use for research without any restrictions. We don't patent them, and we provide the protocol for making the synthetic route for making the compound to commercial vendors so that they can, so that you can buy them from a commercial vendor. And in certain cases, like the newest ones that aren't yet commercially available, we can supply from a request on our website. So if you see anything you're interested in today, have a look at our website or send me an email and if you want to use any of these compounds. So most of our inhibitors are chemical probes. We've published individual papers over the years, but these two resource papers kind of summarize on a large scale about a decade of work. So in our Toronto group at the SGC, we've focused on protein methyltransferases, particularly lysine and arginine methyltransferases. And that collection of inhibitors and a lot of details about them is shown here. So you can read some details here. And my colleague Stefan Knapp, who was originally at the University of Oxford and now is at Frankfurt University in Germany, he and colleagues made a number of inhibitors to bromodomains. And so these two collections are the majority of our chemical probe collection. And so these are resources also for you to have a look at. So I'm going to focus on methyltransferases for the rest of my talk. This is the area I know the most well and have the most experience in. So on the left here is the phylogenetic tree of the so-called set domain lysine methyltransferases. They all have a similarly structured catalytic domain. The little squares show proteins for which we've made chemical probe quality inhibitors. So these all take S-adenosylmethionine as the methyl donor cofactor, and they can either monomethylate, dimethylate, or trimethylate lysines. And the various enzymes have different capacity for these monodi and trimethylation. And then there are reader proteins in the proteome that recognize the difference between mono, di, and trimethylation, and help when they see that signal, they can help assemble a series of protein events on the chromatin to regulate transcription. The other family on the right, so this is a structural family with a different 3D structure, beta barrel-like structure, contains both arginine methyltransferases, so protein arginine methyltransferases, PRMTs, and some similar fold proteins that also methylate on lysine, including DOT1L is a chromatin methylation protein. The other ones are probably not involved in chromatin. And so here, arginine methylation, which I'll talk about quite a bit actually in the talk, it comes in multiple flavors as well, which do different things. You can have monomethylation on the side chain. You can have asymmetric dimethylation, shown in the middle, and symmetric dimethylation, shown here on the right. And so again, these are different proteins that carry out these activities. PRMT5 in red is the symmetric dimethylation. The others do asymmetric, and they can all do monomethylation. So when we started this work, there was excitement about the methyltransferase enzymes because it was thought the analogies were made to protein kinases that take another adenosine-containing cofactor, ATP, and transfer in this case a methyl instead of a phosphate to a substrate, and many of the kinase inhibitors are ATP mimetics or bind in the ATP binding site, and so it was thought that compounds could be found that bind in the SAM binding site of methyltransferases. We have identified a few, but not very many. And what we realized over the years is this is because typically the SAM binding site SAM is a lot more hydrophilic than ATP, and it's the binding pockets that bind to it are also hydrophilic to form hydrogen bonds to it, and it therefore doesn't have as good a drug-like properties, druggable binding pocket properties as we would like. And so only in a few cases have SAM competitive inhibitors been identified. Unexpectedly, more often we were able to identify substrate competitive inhibitors. So these are compounds that bind in the lysine substrate binding channel, both where the lysine side chain goes and also where the peptide, histone peptide would normally be. And I'll show you a structural example in a moment. But we also found allosteric mechanisms of regulation. Arginine methyltransferases are dimers, and so you can, some compounds can disrupt the dimer interface. Many of these proteins are multi-protein complexes, particularly EZH2 and EZH1, which are part of the polycomb repressive complex. In the MLL complexes, mixed lineage leukemia complexes, require certain subunits, and you can, we have identified inhibitors that disrupt the protein-protein interactions within those complexes, and then the enzyme is no longer active. And then finally, I won't have time to talk today, but we've identified compounds that can compete off not the catalytic domain, but other reader domains that are within the protein that anchor it to chromatin for the NSD2 and NSD3 methyltransferases. So there's many ways to skin a cat and many ways to inhibit a methyltransferase. So this is to give you an overview of how we think about, where's my cursor, this as structural biologists, and then use that to think about how to, whoa, what happened? How to inhibit methyltransferases. There we go. So here's the SAM cofactor in the pocket. That's the site of methyltransfer right here in the middle. This is not going. Sorry about this. I don't know why it's taking so long. Oh, it jumped ahead. Oh, sorry. I can only see it on here. Okay, that's, oh, I apologize, it's not, okay, well, I can just do it like this, take snapshots. Oh, now it's moving, okay. Okay, I see what's happening. Okay, so let's do another one. Okay, so here, now we're looking at the peptide binding site, and the lysines going down behind the plane there into the channel. And you can see that the protein is recognizing side chains adjacent to the lysine and recognizing the sequence in which the lysine is anchored. And now you can start to see the SAM again on the back side. Let's jump ahead. Here's a close-up of the substrate peptide. You can see strong recognition of the upstream and downstream side chains because these are very sequence-specific enzymes. Some more of that. Some more of that. Okay, now here's an overlay of our first inhibitor of this enzyme, G9A. And you can, so that's in green. And in the next, now you will see, I believe, the overlay of where the inhibitor binds compared to where the substrate peptide binds. And if we zoom out a little bit, now you can see, so they're still overlapped, and now you can see the cofactor here. That's the sulfur, site of methyl transfer. And you can see that this inhibitor and many of the substrate competitive inhibitors, so this is substrate competitive, it's going to compete. They obviously can't bind there both at the same time, so this competes out the substrate. And it's a little bit close. In this one, it's a little bit close to the substrate, but as you'll see, many of the others are even more close to the cofactor. Okay. So as I mentioned, these can recognize the peptide sequences as shown here in 3 different enzymes. We aligned that methylated lysine upstream, downstream are recognized, and these very selective and specific pockets can now accommodate very different types of compounds that are selective for that given pocket. So the point being, selectivity in this protein family is relatively easy to achieve through the substrate binding pocket, as opposed to the kinase inhibitors, for example, that all bind the very conserved ATP binding pocket where it's hard to achieve selectivity, okay? They're probably not as druggable as kinases because we had to screen a lot of compounds to find even weak hits that would have some inhibition. But when we did find even a weak hit, they tended to be very selective. And so by the time we collaborated with our chemistry collaborators in industry and some in academia and found potent selective compounds, this is the kind of selectivity profile we have so along this axis are the inhibitory compounds, along this axis are human methyltransferases, and most of these you can see are very selective for a single methyltransferase. In a few cases, we have this one, for example. Can't align it. This one can inhibit multiple methyltransferases, but usually fairly selective for a type of family. The other things that we find with these type of inhibitors, which is not commonly seen in enzymes, it's because of the nature of epigenetics, is that the inhibition of a methylation event often takes days to manifest at the lower level of methylation on the chromatin in the cell. And so this is a 7-day experiment where you can see this EZH2 inhibitor reduces the methylation, the trimethylation at lysine 27 in a dose-dependent manner. But we really don't see this at day 3 and day 5. We don't see this mark going away until about 5 to 7 days. And when we look at phenotypes for this inhibitor, it's actually post 7 days that we really start to see the best effects. And you can see that a little bit of reduction in the dimethylation, but the monomethylation, John will appreciate this, is still there. And this is for a different enzyme also. You can see that you get greater reduction with multiple days. And that's because you're inhibiting new methylation going on the chromatin. Chromatin already has methylation on it, so you have to wait for those methyl groups to turn over, either through a demethylase or through cell division. Okay, the other thing we learned about methyltransferases, as I alluded to in that failed video, is that often the cofactor, which is shown here in white with a little bit of purple where it starts to interact with a compound, often the inhibitor in brown here, comes very, very close and actually interacts with the substrate. So a lot of times we have substrate SAM-dependent inhibition of a substrate competitive inhibitor. And this is shown here in this SPR trace. And the extreme example is this PRDM9 inhibitor that you can see is just nestling right up against the SAM, including the adenosyl ring here. And so, it was kind of 7-8 years before we really realized this trend. If we had to go back now, we would really, really incorporate the cofactor very much into the sort of design of these inhibitors. And I think going forward for scientists and medicinal chemists to do so is it's a new, I think it would be a even more fruitful strategy. So this is our collection right now of epigenetic compounds, chemical probes. So we have the target classes in different colors, the name of the protein, and the names of the chemical probes. They're often named for the company or the research lab that made them with initials. And I've highlighted here those protein targets outlined in yellow for which there are either always a different compound, so a different drug, in a clinical trial or a late-stage preclinical development going on. So that, it's maybe a quarter or third of these targets. So there's a lot of interesting translational biology that can be done here using even just our chemical probes in cellular disease models and in some cases in vivo models to try to understand what types of cancer cells or other disease cells are amenable to inhibition of a given target that would give the desired modification to the disease phenotype. And so that's what we've been doing a lot of, and that's what I'm going to present in the rest of the talk, is a couple stories or vignettes along those lines. So we have been collaborating with colleagues who work on glioblastoma, pediatric cancers, colorectal cancer, poor prognosis AML samples, and clear cell renal carcinoma, and breast cancer, which I'll show in a moment. But so this shows an overview of this work, and you can see that in some cases, like inhibition of BET bromodomains, compounds like JQ1 stop growth of most cancer cell growth. Other inhibitors, like EZH2 inhibitors, are effective in some context, right? So in colon cancer, I'll show in a minute, it stops growth of all these tumor cell spheroids. And then other samples are effective only in certain contexts. And so we're very interested in what are the genomics and epigenomics like in patient-derived samples that respond to a given probe, and then those samples that don't respond to a given probe. And so these are all patient-derived cells we're using here. So the first example was in colorectal cancer in collaboration with my colleague Catherine O'Brien and carried out by Evelyn Lima Fernandez, a postdoc in the lab. So just for those of you who are not familiar with it, EZH2 is the catalytic subunit of the polycomb repressive 2 complex. It trimethylates lysine 27 on histone 3, and that is a repressive mark. It leads to the chromatin compaction and repression of expression of genes in there. If you inhibit EZH2 over several days' time in cell division, the chromatin will become de-repressed, and you'll get more looser chromatin, often with other gene-activating mutations taking over. So in this set of experiments, Evelyn was first working with, we were interested in trying to target this cancer stem cells in these colon cancer tumor organoids that Catherine's lab had developed. And we noticed that Evelyn did a screen of all our probes and found EZH2 inhibition was growth suppressive, strongly growth suppressive in the tumor organoid model, but not when you grew the exact same cells in sort of normal suspension media. The cells would become differentiated in that case and just didn't care about EZH2 inhibition. But we think that the tumor organoids are more representative of a real tumor. And so what Evelyn found is what I just said. It suppresses these organoids. And it also, in limited dilution assays, decreased the frequency of colon cancer initiating cells. And if we do epigenomics, this is ChIP-seq of the repressive mark that we're inhibiting with EZH2 and an activating mark that is associated with gene activation and transcription. And in certain cases, you have these two marks at the same loci. This is called a poised locus that is poised to become expressed, perhaps, for example, during development when this repressive mark is removed. And what we found in our epigenomics is that the Indian hedgehog locus, which is a major regulator of colonic differentiation, was in this poised state. It was this is probably one of the things that's keeping it in a stem-like state. And so when we inhibit with EZH2 inhibition, this repressive domain goes away and it's ready to express. And we showed that we could phenocopy the phenotype of EZH2 inhibition with recombinant Indian hedgehog protein. And this has been published for a couple of years, so I won't go through all the details, but it does look like EZH2 inhibition would be effective in colorectal cancers, probably in combination with chemotherapy. Glioblastoma is another area that we work in with Peter Dirks, who's also interested in cancer stem cells. And in the screen that postdoc Patty Sackamiter did with his samples, these are well-characterized genotyped samples, we found that PRMT5 inhibitors, results shown along here, were effective at growth suppression in GBM. And we have two types, substrate competitive and a cofactor competitive inhibitors. So two mechanisms of action that we can use side by side, and then an inactive control compound that's very similar between the two, shown here. And so now this is arginine methylation, and PRMT5 is the major symmetric dimethylation enzyme here. It's kind of grouped in with epigenetic regulators because histones are a target for these enzymes, but a more abundant set of targets are RNA binding proteins, and particularly those involved in the splicing machinery. And so what I'm going to end up telling you about now is a splicing story. But first, to the phenotype, we see PRMT5 reduces the sphere-forming capacity in patient-derived GBM cells. And this is shown for both, so we get pretty much the same result for both compounds. And it's related or correlated with reduction of the symmetric dimethyl mark. This is the nice thing about methyltransferase inhibition biology. You have a very good biomarker to tell you that your compound's getting in the cell and doing what it's supposed to do. And we also see, and so this is also sphere-forming capacity inhibition, so it's affecting the stem compartment. And then it promotes senescence and apoptosis in these cells that are no longer growing. And if we look at these growth curves here for 4 different patient-derived models, you can see that particularly those with the more potent Lilly compound can have a very sustained growth suppression that doesn't come back even after we stop treating. That's the dotted line. When you stop treating, in certain cases, the growth never comes back. So that's encouraging. So we wanted to know, but not all the samples. So there's I think almost 30 some, maybe 40 patient samples shown here, and we get a gradation of responses to the inhibition, as any collection of biological samples tends to do. So we wanted to understand what was the difference between the non-responders and the responders. And so we looked at the major known mutations that occur in GBM, shown here. We looked at the TCGA classification. And it does look like there's some segregation with the proneural classification, shown on the right. And then we also, another putative target in the literature was the methylthioadenosine phosphorylase gene, which is coincident, but in the opposite sense, to CDNK, this P16. This is P16 tumor suppressor. But so this locus is often homozygously deleted, and you often get no MTAP protein expressed, but as you can see, it doesn't really cluster. So MTAP deletion per se does not tend to cluster with response or not. But why are we looking at MTAP? I just want to show this for a minute, and that's because it's a key enzyme in the tryptophan, the methionine biosynthetic cycle. And when MTAP is deleted, you get a buildup of methylthioadenosine. You can see it looks like SAM. And when you get a buildup of that oncometabolite, it inhibits PRMT5 and not the other protein arginine methyltransferases. So this was, there were a number of publications saying that PRMT5 is going to be effective in MTAP-deleted cancers. And it is, but it's also stops growth in other cancers, in non-MTAP-deleted cancers. And it actually turns out to be a pan-essential gene in cancer, and it stops a lot of growth in various cancers. But in ours, we get this range. So we wanted to understand what the difference was. And this is where we turn to RNA-seq and notice the splicing. So when you do RNA-seq, consistent with the phenotype, we see enrichment for cells, proteins involved in DNA repair, cell cycle, and splicing. When we look deeply at the splicing effects, we see a large number of retained introns. Now, this is probably because PRMT5 methylates a lot of proteins involved in RNA binding, particularly those in splicing factors, and probably compromises somehow the spliceosome. We don't know exactly how yet, but probably in some sense. And so then if we take all these retained intron proteins and predict the negative impact of the alternative splicing events, you see that those cells with those proteins with a negative predicted impact are all involved in cell cycle. So I think we have a fortuitous block to cell cycle and subsequent apoptosis or senescence involved. But it still doesn't explain why some samples are responsive and others are not. And it's not drug transporters. We looked at that. So the only thing we could identify was a pre-existing splicing pattern pattern that differed between the sensitive and the non-sensitive cells. And some of those proteins that were in this differential gene set were those involved in cytoskeleton and centriole. And this caught our eye because of this funny phenotype that we saw in the cells when you treat them with PRMT5 and then they stop growing or they die. You get these funny nuclei that are like bilobal or circular, and we think that they're trying to divide, but something's happening. The right proteins are not there anymore, and it can't completely divide. So I don't know if anybody's seen this before, but if you have, maybe at reception we could talk about it because we're interested in what's happening here in the cell. Okay. So, and we did in vivo orthotopic experiments showing that PRMT5 inhibition could reduce the tumors in the mice and provide a survival benefit. And again, you can show that the drug was working or the chemical probe was working because you got reduction of the symmetric dimethyl mark in the tumors in the mice. I won't go, due to time, I won't go over the summary part, but I just want to say relevant to translation to the clinic, the first generation PRMT5 inhibitors, sadly, such as those that are similar in chemical structure to the GSK inhibitor that we used, have proven to be too toxic in clinical trials, and several companies have stopped their phase 1 clinical trials. The thought is that that second generation PRMT5 inhibitors are being made that will take advantage of PRMT5's ability to be inhibited by methylthioadenosine in the context of MTAP deletion. And so there are a number of companies working on this right now. As we showed, it's a common occurrence in GBM, so at least half the patient samples would have deletions of MTAP and might be amenable to this approach. And I think we have to wait for those kinds of compounds to be made to see if this would work. But I think it's interesting that even by using the first generation chemical probes and testing it out and showing that in the case of this wide breadth of samples that we could identify a proof of concept, or just the therapeutic window was probably not wide enough. And okay, in the last few minutes, I'll go through the last example where former postdoc who's just left to go back to and start her lab in China, Qing Wu, and my grad student David Ni have worked on the case of type 1 PRMT inhibition in breast cancer. And this was a collaboration with Mathieu Lupien, Benjamin Hadkaynes, and Dave Chescon. So what Qing did was take this series of triple-negative breast cancer cell lines that Mathieu had compiled in his lab and had RNA-seq and genome-wide analysis and attack-seq for. And she did a screen with our chemical probes shown here. And what she found was that this compound MSO23, which is a pan-type 1 arginine methyltransferase inhibitor, was effective in some cases, the red squares, but not others. And she was very interested. She published a couple other nice papers I don't have time to talk about. Again, figuring out what is it about certain cell types that make them sensitive to one drug and not to another. So she set off to figure out what made things sensitive or not to PRMT1 inhibition, because pan-type 1 PRMT inhibitors were at the time also in the clinic by GSK in a number of diseases, I think including breast cancer. So what we see, it's a type 1 PRMT inhibitor, so this compound inhibits all these enzymes here and inhibits this asymmetric dimethyl mark. We classified the cell lines according to either red for responders, those that were sensitive to inhibition and didn't grow anymore, versus those that did not very well, so going into the black here. And the inactive control compound, I always like to use our inactive control that's chemically similar but doesn't inhibit the enzyme as a chemical control. And you can see that in both cases, the sensitive and the non-sensitive are both, the methyl mark goes away in a dose-dependent manner, so the compound's getting in the cell and inhibiting the target, yet some stop growth and the others don't. So what's going on? So first we wanted to see which of those methyltransferases what it was, and we have now selective inhibitors to PRMT4, 6, 3, and 4, and none of them have any effect on the protein, just MSO23. So we deduce that it's probably PRMT1 being inhibited that causes the growth suppression. If you look in DEP-map, a cancer dependency map, PRMT1 is a pan-cancer, or is an essential gene in triple-negative breast cancer. In mouse models, we could show that PRMT1 knockdown was growth suppressive, and treatment with MSO23 would phenocopy this, shown here. And again, we can track the mark is being reduced, so the compound's getting to the tissue in question. And we also looked in tumor organoids that were sensitive or resistant to MSO23, and you can see that this extends to direct patient-derived samples as opposed to cell lines that have been passaged many times. So what is it that's different about the cells that respond to MSO23 and those that don't? And so we go to gene expression analysis, and you can see this. So this is without treatment. The cells that the differential expression versus responders responders versus non-responders leads to a lot of signal from interferon signaling. There's something going on with innate immune signaling here. And this just shows that we can see the same thing in patient-derived tumor organoids between the responders and the non-responders. So now if we treat the cells, the responsive cells, so after treatment, we get a further increase of this interferon response signal. And so this figure here in the middle shows no treatment and treatment. And you can see with the treatment, we get now upregulation of a lot of interferon response genes. And these were validated by qPCR. And so this PRMT, it's PRMT inhibition. We therefore also looked at splicing. And so we analyzed the splicing events, and we can see that there is a large number of retained introns. And right about the same time that we were mulling over this, what's going on here, another paper came out in Cell, Bowling et al., showing that inhibition of splicing kinases could induce a double-stranded RNA and a viral mimicry response, double-stranded RNA in the cytoplasm through missplicing events. And so we thought we would look there. And we turned to my colleague, Daniel DiCarvalho, who really specializes in this area of viral mimicry. And he said, well, look at your introns that are retained and see if they have inverted repeat sequences, these ALU repeats. And sure enough, at least half of these retained introns do have the potential to form double-stranded RNA, which could go to the cytoplasm. And if you stain for double-stranded RNA, we see it in the cytoplasm. Okay, and this is quantification of that. And so we think we're seeing a triggering of an antiviral immune response here with PRMT inhibition through missplicing, which generates RNAs now that can form double-stranded RNA. And this just shows that the receptors that recognize double-stranded RNA, this is showing knockout of various receptors, and we see signal for RIG1 and TLR1 shown here when we combine it with MSO23 and knockdown, you can get a rescue. So in summary, what we see here is that we can treat the whole series of cells with type 1 PRMT inhibitors. We reduce the methylation on arginine, particularly in splicing factors, we're assuming. We haven't directly tested that. But we have a different transcriptome in the two cell types. The unresponsive cell types have a transcriptome that does not have the potential to form these inverted introns, or if they're formed, they're not being transported to the cytoplasm, and you just get you get missplicing of the RNA, but it's not immunogenic. Whereas the sensitive cell lines have they have the potential to form these retained introns with inverted repeats, and you get this immunogenic signal. And then if you recall, at baseline, the insensitive cells did not have a strong interferon signal. The responders already had somewhat of an interferon high signal, and now you increase it even more. And so we think this pushes the cells over the edge, over a threshold in which they die. And this is very akin to what this paper by Bowling et al. Had observed for using splicing kinases. And so Daniel's group wrote a review of this paper, the Bowling paper, and I think we're seeing a similar thing. We have a pre-existing cancer-specific, low-level interferon signaling expression program. So low level, low enough for the cells it's there, but the cells can survive with it. And then when you enhance this response beyond a tolerable threshold with PRMT inhibition, in our case, the cells will die. And interestingly, Daniel pointed me to this paper. It was in bioRxiv. I haven't checked to see if it's published, but showing in a vast genome-wide analysis of inverted repeats and other repetitive elements that these repeats mimic immunostimulatory features across a vast evolutionary landscape, so that somehow the genome encodes these sequences that are potentially immunogenic and bad for the cell when released, but they're in there and they are conserved in our genomes. It's not just an accident of a virus. It's some reason they're there. And maybe we can take advantage of it for cancer therapy. Okay, so I'm just about out of time, so I'm going to skip through this just to say that multi-domain or multi-subunit proteins harbor these reader domains, these so-called domains that recognize methylacetylysine. Many of them are druggable. We have made chemical probes to many of them. And now with the advent of the drug modality of PROTACs, or targeted protein degradation, you can also use these as sort of handles onto the protein of interest to degrade the entire protein. You can do that to the inhibitors as well, but this gives more opportunities for targeting different parts of a protein of interest for therapeutics. And so I'm going to end back to my question of, how do we drug the entire proteome? And this relates to the question of precision medicine. We all would like to do precision medicine and have drugs to do precision medicine, but what that requires is a large pharmacopoeia of medicines for the doctor to choose from to treat a patient, and to know which drugs go with which personal or specific or precision aspects of the patient's disease. And we're very far from having those tools available. But if we had more pharmacological tools to all the proteins in the proteome, we could start to get there. And so we've initiated this initiative. We can't do it ourselves. No individual or group can do it themselves, but we would like to stimulate or encourage the community to think about, as you go about your research, or even purposely go towards this goal, and develop technologies, and if you're making an inhibitor, a chemical probe, provide it to the community to use, and join the effort, if you will, to try to identify pharmacological modulators to the entire proteome. And we think in this current first 5 years, this is 2 years old from when we published the paper, the first 5 years, technology development will be very important. And so, if you have technologies that will accelerate the rate at which people can identify small molecule inhibitors, there's some tools on the website, our Target 2035, to make introductions to different groups with complementary expertise. You can check it out. Okay, so I think I'll end there. Time is running out. I just want to thank my many, many collaborators and colleagues at the SGC. I presented a vast amount of work over 10 years from many, many people. Particularly, I want to cite Peter Brown, who helps coordinate our entire epigenetic probe program, Mathieu Shapira for computational aspects, Jinrong Min, structural biology, Masoud Vedadi, enzymology, Dalia Barsite's cell assays, and Takis Prinos worked on the Stand Up to Cancer project with Peter Dirks' group on glioblastoma, my trainees in my lab and my collaborators at Princess Margaret Cancer Center, the other SGC labs. And in particular, I want to thank all our chemistry colleagues at the pharma companies and academics who make these compounds with us and make them available to others to use with no strings attached, and provide the physical compound to actually give out to people. So I'll end there. I will say we have faculty positions at Princess Margaret Cancer Center for protein science, chemical biology, structural biology, anything in that realm, proteomics. If anybody's starting to look for faculty positions, you can give me a shout. I'd be happy to talk to you about it. Thank you. Okay.
We have some time for some questions. Anyone like to start?
Thanks. So I was interested in sort of whether you could it seems fascinating how different cancers have different sensitivities and that your drugs are exquisitely able to start to get at those sensitivities. And so with the last story, can you test this by knocking down, say, RIG-I or something, one of the sensors, and showing that it's directly acting through the immune sensing response? Or is there other ways to target this to show, like, could you upregulate it in the non-sensitive and show that they're now sensitive? Things like that.
Yeah. I don't know, at the receptor, well, at the RIG-I receptor level, upregulating in the non-sensitive, I don't think would work because we're not getting the double-stranded RNAs there, we don't think, right? We haven't been able to measure that. Can we knock, I think we did some knockout. I think we knocked down, we might have, we knocked them down, right? Yeah, that's what we did. That was the slide I went over the end. It was really quick. Yeah, we knocked each one out, and then, and they were dependent. Yeah.
I had a question about the difference between the PRMT1 and the PRMT5 story that you talked about just before it. And while the MTAP deletion didn't seem to correlate very effectively, but at least on some substrates, PRMT5 and PRMT1 work on the same arginine, one doing asymmetric dimethylation, the other symmetric methylation. I'm just wondering, in the Peter Dirks collaboration, did you look at PRMT1?
It was in our initial screen, and we didn't see a strong signal there. We didn't look at combinations, but there's a lot of interest in. Well, for a time, there was a lot of interest in combining PRMT1 and PRMT5 inhibition. But I think they both don't, at least PRMT5 doesn't have a very good therapeutic window, so I think those studies, you do get more of an effect with the two together. But PRMT5 is really kind of more powerful than, it seems like, in terms of growth suppression and cell death. So I know there's Ernesto and colleagues in New York have published on the combination in leukemia, right? Yeah. Mm-hmm.
Cheryl, it's great to have you here, and particularly for this occasion. So thanks for coming and giving a great talk. So, back at the PRMT1 inhibitor, so a nice readout looks like histones were a great readout for loss of. So you could phenocopy with a knockout or chemical treatment. So I'm just curious, what's your next strategy to figure out what is the actual substrate if it's within the splicing machinery? How are you going about that, say through a chemical biological approach, or doing sets of kinds of things where you're denaturing cells and looking to see who sticks around with and without methylation? I mean,
we haven't really explored what the substrate.
Give us the secrets, please.
Sorry, we haven't explored what the substrates are really, because we're not a splicing lab, or we're a spliceosome lab, right? I think that's, I mean.
Are some of your colleagues doing, chemical, making deriving the inhibitor in a way that you could do a proximity ligation experiment or something like that, so you could try to?
Well, it's well known that the PRMTs, PRMT5 in particular, methylates RGGR, these repetitive arginines with glycines sequences, which are very abundant in RNA binding proteins and in splicing factors, and so what it means is that you need methylation of those sequences to properly assemble a spliceosome, presumably, or some function of the spliceosome. But it's just, out of my wheelhouse at the moment. So any, collaborators who know about splicing would be welcome.
Great, thank you.
Any other questions? All right, so we have a reception outside, so please join us all out there. And again, join me in thanking Dr. Cheryl Arrowsmith.
Thank you.